Okay, let's get started. So welcome everyone to the SADS Foundation's Living with SADS webinar on gene therapy, ACM, PKP2 clinical trials. I'm Marcia Baker, the program director for the SADS Foundation. We're honored tonight to have Dr. Andreas Barth of Johns Hopkins, who is volunteering his time and expertise for this webinar. On behalf of the SADS Foundation, we thank Dr. Barth, as well as our industry participants who are sponsoring ACM PKP2 clinical trials and this SADS webinar. We appreciate the support from these three companies. In alpha order, they are Lexio Therapeutics, Rocket Pharmaceuticals, and Tanaya Therapeutics. The SADS Foundation is grateful for your participation today to educate the SADS community and address their questions in the breakout rooms. Here's an overview of the timing for today's webinar and how it will be structured. After Dr. Barth's presentation and question and answer session, we'll hear from each company's medical representative with a 5-minute overview of their company and its clinical trial. Following the company overviews, we'll break into 3 separate Zoom rooms where you'll have the opportunity to ask questions of each company, 20 minutes in each breakout room. So please hold your specific clinical trial questions for these breakout rooms with the companies. A reminder that only information that is in public domain and on clinicaltrials.gov will be shared today. As an attendee, you will stay in your assigned breakout room while the company representatives will rotate so that you can ask questions of each company. It's now my pleasure to introduce our speaker, Dr. Andreas Barth. He is a physician scientist and medical director for the Center on Inherited Heart Diseases at Johns Hopkins University in Baltimore. As an adult electrophysiologist, he has expertise in inherited arrhythmias and SADS conditions. Immediately following Dr. Barth's talk, you'll have the opportunity to ask him general questions, not related to a specific clinical trial. Your questions for Dr. Barth may be typed in the chat following his presentation. So thank you again, Dr. Barth, for your time tonight to talk about these gene therapy ACM PKP2 clinical trials. I'll turn it over to you to share your screen and your PowerPoint now.
Thank you so much for the kind introduction, Marcia, and thank you for giving me the opportunity to participate today in this very exciting webinar. Let me see whether I can share my screen. Put it on presenter mode.
That's perfect. That's it.
Yeah?
Mm-hmm.
Good. Fantastic. So I believe this will be a very valuable experience for the ARVC community to be able to ask questions and to be directly engaged with the companies involved in gene therapy. So my disclosures are that Johns Hopkins will be enrolling for 2 of the 3 companies, for Lexio and Tanaya, in PKP2 related gene therapy trials in the second part of 2024, and I'll be the site PI for those studies. And I'm also on the advisory board for an additional gene therapy company, Solid Biosciences, that's unrelated to PKP2. So I structured my talk in 3 different parts. So first, what is gene therapy? The second one is, how does gene therapy for ARVC work specifically? And the third one is, who is it would be eligible for PKP2 related gene therapy? Regarding the first aspect, what is gene therapy? I would like to refer to 2 seminal webinars from SADS that are actually available on the SADS website. It's from my colleague, Andrew Landstrom, who's a pediatric cardiologist and a genetic cardiologist, who did 2 incredibly outstanding webinars in the past. One was just very recently, more the basics about gene therapy. And there is an older webinar from last year that's still very, very up to date and very recent about gene therapy. And he also covers some specific aspects about ARVC in that webinar as well. So just to recap, what is gene therapy? What are we really talking about when we are referring to gene therapy? Gene therapy uses genetic material, DNA, to treat or prevent a medical condition. And we can use gene therapy to replace a gene that is causing a medical problem. We can repair a gene so that it's no longer causing a medical problem. We can turn off a gene that is causing a medical problem. And last, we can add a gene that helps the body to fight a specific disease. Today, when I'm referring to gene therapy, I'm referring to gene replacement therapy. So how can we replace a gene that is deficient and the deficiency is causing a medical problem? I want to point out that gene therapy is commonly used in cancer mostly. There are more than 2,600 active gene therapy trials in cancer. And in cardiovascular diseases, it's still a relatively early field. And there are less than 5% of all gene therapy trials are involving cardiovascular indications. However, this is rapidly growing. And currently, there are gene therapy trials for hypertrophic cardiomyopathy and dilated cardiomyopathy. For ARVC, there are some gene therapy trials for heart failure associated with muscular dystrophy and for Friedreich ataxia. And very important for SADS is that there are also gene therapy trials on the horizon for conditions like long QT syndrome and CPVT. And the future has already started. Last year in October, the first patient was enrolled in a human gene therapy trial for hypertrophic cardiomyopathy. And I'm very sure that we'll see many, many more in the near future. So how does gene therapy specifically work for ARVC? So it's important to understand what is ARVC. So ARVC stands for arrhythmogenic right ventricular cardiomyopathy, which is a very complicated long term. And arrhythmogenic means that it's related to irregular heart rhythms, and cardiomyopathy refers to a disease of the heart muscle. So ARVC is a condition where the muscle in the right ventricle of the heart gets replaced by fatty or fibrous tissue, making it weaker and more prone to develop irregular heartbeats. So think of your heart like a house with different rooms. The right ventricle is one of those rooms responsible for pumping blood to your lungs to get oxygen. When someone has ARVC, this room starts to deteriorate. Instead of being strong muscle tissue, it becomes fatty or fibrous, kind of like the walls of the room getting weaker and thinner. And because of this weakening, the heart can have trouble pumping blood properly, and irregular heartbeats can occur. So these irregular rhythms can be dangerous and sometimes lead to more serious issues like fainting or even sudden arrest. And the most common gene mutation leading to ARVC is found in a gene called plakophilin-2 or PKP2. So to better understand the role of PKP2, we need to zoom into the heart a little bit more. And if we zoom into the right ventricle and are able to examine this under a microscope, we see that heart tissue looks like individual cells that are connected to each other. And this creates a network of cells where individual cells merge to function as a unit. And in the heart, this is crucial because it allows the cells to coordinate their actions very closely. When one heart muscle cell receives a signal to contract, it passes the signal rapidly to its neighbors. This means that the cells can be together in a synchronized way, which is essential for pumping blood efficiently throughout the body. And these squiggly lines that you see here are the cell borders. They connect cells individually. And if we zoom in a little bit more, you can see that in these cell borders, you find structures which are called the desmosomes. So imagine desmosomes as tiny buttons that hold neighboring cells together like snaps on a jacket. They're like strong adhesive patches between cells, and they ensure that cells stick together tightly. And these structures, these desmosomes, are essential for keeping tissue strong and intact, especially in places where cells need to stick together very tightly, like your skin or the heart muscle. So basically, these desmosomes are like the glue that holds cells together, helping to maintain the structure and stability of these tissues. And PKP2 or plakophilin-2 is an essential part of this desmosome. So if you lose plakophilin-2, then this glue gets weaker and the cells detach from each other. So we know that plakophilin-2 is a component of the desmosome, so helping the cells stick together. In addition to cell adhesion, PKP2 is also crucial for controlling the levels of certain genes that manage calcium inside cells. Calcium is vital for heart cells to function properly, so it helps the cells to contract and relax. And this specific study showed that when PKP2 is missing, the production of several important proteins that handle calcium is impaired. And this disruption in calcium management can lead to irregular heartbeats or arrhythmias, even if the heart structure looks relatively normal otherwise. This is another very important study that asked a very important question. How many heart cells do we need to correct with gene therapy to see a therapeutic effect? And you can imagine when if we want to correct the pump function of the heart that is weak, we really have to improve or achieve gene expression in the majority of the heart muscle cells to improve heart function. When we think about preventing cardiac arrhythmias, however, we may not need to transfect or transduce all the cells in the heart to achieve a therapeutic or beneficial effect. And using computational modeling, this study looked at tissue under a variety of conditions to test the ability of genetically engineered stabilizer cells to suppress triggered arrhythmias or PVCs. So here you see 30 cells that are coupled together electrically and give rise to an action potential. And in disease states, these cells give rise not only to a first beat, but to PVCs, so to an extra beat. And if you have multiple of those PVCs in a row, you create ventricular tachycardia, which is the dangerous heart rhythm in ARVC. So in this computational modeling studies, about 20% of the diseased cells were replaced with healthy cells. And the diseased cells are shown in red, and the corrected healthy cells are shown here in black. And because the cells are electrically connected to each other, the healthy cells were able to suppress the PVC. So by introducing the gene only in a minority of cells, in 20% of cells, this study showed that you're able to suppress arrhythmias effectively. So how are we able to introduce PKP2 into the heart muscle? So we could take a cell and just inject DNA or PKP2 into the naked DNA of the patients. However, we know that this is really not working very well. So this is really not done. And we found a more efficient solution, and this is namely to package the PKP2, the gene of interest, within a viral vector that increases the efficiency of gene delivery. And the viral vector that has emerged as a suitable virus is called adeno-associated virus, or I refer to that as AAV. AAV is used in a wide variety of gene therapy applications today. So we use it in gene therapy for central nervous system diseases, for spinal muscular atrophy, for certain forms of muscular dystrophy, but also for gene therapy that involves the eye, or for certain metabolic diseases or lysosomal storage diseases. So it has really emerged as the viral vector of that is preferred in many studies. And why is it preferred? What is the advantage of using AAV? So first of all, it's non-pathogenic, which means that infection with AAV is not associated with disease in humans. And it's replication deficient, so it won't be able to replicate on its own. And while it creates or elicits an immune response in the body, the immune response is actually relatively mild and relatively minor. Additionally, with AAV, you're able to induce a long-lasting gene expression in cells. And very important when considering gene expression for the heart, you can infect both non-dividing and dividing cells. And this is important because cardiomyocytes or heart muscle cells are terminally differentiated, which means that they're not dividing actively. And importantly, there's a low risk of genomic integration. With other viral vectors, they can randomly insert the genes into the genome, And there is a concern that you may disrupt some genes that are critically important for the function of the cell and the organisms. So, for instance, if you disrupt certain tumor suppressor genes, you may induce cancer, and the risk with AAV is really minimal or exceedingly low. So, with all those benefits, there are also challenges when it comes to AAV therapy. And most importantly, many people have been exposed to different forms of AAV in their lifetime, leading to the development of antibodies against the virus. These antibodies are part of the immune response or immune system's memory and are designed to quickly recognize and neutralize the virus if it enters the body again. So if a patient already has antibodies against AAV, these antibodies can bind to the AAV vectors used in the gene therapy. And when this happens, the immune system sees the virus as a threat and destroys it before it can deliver the therapeutic genes to the cells. So additionally, transgenes remain transcriptionally active indefinitely, which means that we are good at introducing the gene into the cells, but there's really no off switch. If we don't like what we're seeing, there's really not an easy way back. And gene therapy is also limited by the small packaging capacity of AAV. So not every gene is suitable for packaging into AAV. And there are also some patient and caregiver concerns. We really have to manage the expectations and explain what gene therapy is and how close it is. It's often mistaken that it's a cure, but we don't really know whether the effect is long lasting, whether it's years or 10 years, or is it going to be good for a lifetime. And last, affordability will be an issue on a society level. So the first gene therapies that became available for spinal muscular atrophy, for instance, and just to remind you, this is a one-time infusion of this gene therapy, is 2.1 million. So it comes with a hefty price tag. For Duchenne muscular dystrophy, the price is even higher. So there are really affordability concerns with gene therapy. So if we deliver gene therapy, how can we make sure that the gene is making it to the heart? So first, we can select our AAV serotype wisely. So you can imagine that there are different AAV serotypes, which means they come in different envelopes. And different AAV envelopes have a certain tropism, so a certain affinity or preference to a specific tissue. So let's say you're planning a gene therapy for the brain. You will choose, for instance, AAV2, which is quite selective for the brain. However, if you're looking to infect the heart, you may choose AAV9, which is highly selective for the heart. But it's important to know that it's not only going into the heart, but you will see some expression of AAV9 also in muscle, in the retina, and in the lung. So how can we further improve the tissue selectivity? So there's this concept of AAV expression per set. So what does this really mean? So we told you that the envelope to deliver the gene is the AAV serotype. But inside this envelope, we package the gene. And if we choose a gene that is primarily expressed in the heart, like PKP2, and we put this gene under the control of a promoter, which is the engine that drives gene expression. If this promoter is active only in the heart, we may be able to achieve high tissue selectivity inside the heart. So by choosing the right AAV serotype and the AAV expression percent, we may be able, just by giving a single IV infusion, to have that gene delivered specifically to the heart. Last, I want to focus on some specific aspects about PKP2 gene therapy clinical trials. So who would be eligible for ARVC gene therapy trial? Well, at this moment, we're talking about PKP2 variants only. So if you have the most common form of ARVC, which is PKP2, that gene therapy would potentially be suitable for you. If you have a different form of ARVC, let's say desmoplastic or desmoglian, then this particular form of gene therapy would not work. You have to meet the clinical diagnosis. So not only do you have to have the gene, but you also have to have the phenotype. You have to have symptoms of the disease according to what we call the 2010 revised task force criteria for ARVC. You have to have frequent PVCs. That will be one of the criteria for many of those studies. And importantly, currently most trials will include only adult patients. The regulatory requirements to include children are significantly higher, the burden is significantly higher. The first studies are intended only for adults. You have to have an ICD, so implantable cardioverter defibrillator, and be on stable medication on a regimen for at least 30 days. Some studies may require even longer. As discussed previously, if you have antibodies against AAV, you will not be a candidate for these trials, and you will be tested specifically for those antibodies before these trials start. You have to use contraception for 12 months if you are to be included in these trials, and you have to be willing and able to participate in the long-term follow-up. And this is an important factor that I want to stress. And this slide is important to give you an idea what commitment is required to participate in a gene therapy trial. And this is an example from a muscular dystrophy trial to highlight some of the features that all gene therapy trials have in common. And they are different between individual trials. And on purpose, I did not choose one of the 3 ARVC trials today, because I really don't want to favor one company over the other and give you sort of a false impression. But I want to give you more of a general concept what is required. So I would actually encourage you to discuss specific details about individual trials with the companies in the breakout sessions. I think this will be an incredibly valuable experience. What all those trials have in common is that you need to be hospitalized overnight for at least 2 to 7 days after the gene therapy is administered. More importantly, for about 2 weeks after a gene therapy is administered, you need to stay close to the trial site in order to be monitored for side effects. So you will require a blood draw probably every other day for two weeks after the gene therapy trial is administered to monitor for side effects. These studies are not a study that is done within a week or 2, but you will be required to participate for up to 5 to 6 years. And this particular muscular dystrophy trial had about 49 study visits that were required. Now, most of those study visits happen early on. They happen in the first months, they happen in the first 6 months, in the first year. And after the first year, it gets significantly lighter with respect to how many study visits are required. But you're required to participate in a long-term follow-up study. And that would require every 6 months for up to 4 years to still present to the study site to be evaluated for long-term follow-up. And this is mandated by the FDA if you participate in gene therapy trials. So who is ineligible for ARVC gene therapy trials? Well, if you're unwilling to comply with study procedures, including your follow-up, that's an exclusion criteria. If you have severe heart failure symptoms, so either the right ventricle or the left ventricle, the main pumping chamber, don't work very well or are severely impaired, you may be excluded from those trials. If you're still actively participating in competitive sports, which we know leads to more rapid progression of ARVC, we really don't want you to be included in those trials because this sports participation will be a significant confounder, and we may not be able to assess the effect of gene therapy very cleanly. If you have significant liver or kidney disease, you're not a candidate. If you have active infections, which require systemic antiviral or antibiotic therapy, you're not a candidate. If you have prior infection with AAV, so if you have prior pre-existing antibodies, you would be a candidate. And in the last couple of slides, I would like to highlight some features that the development of drugs have and contrast those with the development of gene therapy trials. So every drug development starts with preclinical research, and then you move on to clinical research. And in clinical research, you have three different phases of clinical trials. You have the phase 1 trials, which are usually done in healthy volunteers, where it's all about safety. And then there is a phase 2, where you look at efficacy. And in phase 3, you usually test it in a larger cohort of patients, where you say, okay, I have patients group 1 where we randomize them to receive the study drug, and the other half of the study population will receive a placebo, so a drug without any active ingredient. And then we'll compare them. Who will do better over time? Hopefully, we'll be able to confirm the benefit. And initially, we start with few participants, and as you move on through these studies phases, you will have more and more people enrolled. And then the drug will be approved by the FDA. In post-marketing, you do safety monitoring. So you get real-world data, what the safety profile and efficacy looks like in the larger community. Now, to contrast this drug development with gene therapy trials, is that in drug development, we have this separation between safety and efficacy. And in gene therapy clinical trials, the safety and efficacy are included, they're combined. And this is important because the efficacy of a therapy, how well it treats or cures a disease, can directly impact its safety profile. For instance, if a gene therapy trial is very effective but causes severe side effects, its overall benefit might be limited. Conversely, a safe treatment that does not significantly improve the patient's conditions may also not be useful. So being able to assess both aspects together helps us to find a balance between the benefits and the risks, and combining safety and efficacy assessments in the same trials allows us to gather comprehensive data more quickly and more efficiently. And this is particularly important in the field of gene therapy, where treatments are often groundbreaking and data on both safety and efficacy is needed to move forward with regulatory approvals. But whatever we do, gene therapy, all the gene therapy trials, safety comes first. And the way we handle it here at Hopkins is that we have a local gene therapy team of experts to monitor side effects of gene therapy. And only if we know that this is safe, there will be a dose escalation. And there is an independent data safety monitoring board to reduce all adverse events that occur during the trial period and make sure that it's safe to proceed with the trial. We really have the most important aspect for us is your safety. With all that said, there are still open questions when it comes to gene therapy trials for ARVC. What we don't know is that what if patients have already pre-existing antibodies against AAV? Are we able to circumvent them? Are we able to navigate find ways around it? What are the optimal dosing for regimens for gene therapy? And we also need to have patients on immunosuppressants. I told you that the AAV have a low immunogenicity, meaning that they elicit a very low immune response. However, if you give a large dose of that virus, that immune response may be actually quite significant. And it's standard of care in gene therapy trials. Again, it varies between different companies. You need to give an immunosuppressive regimen, and that includes steroids, but that includes also at times medications that are given very similar to what is needed if you have an organ solid transplant, like a heart transplant. Now, in contrast to a heart transplant where you require a lifelong immunosuppressive regimen, the immunosuppressive regimen for gene therapy trial is only needed for a very limited period of time, whether that's 1, 2, or 3 months, but usually for the immediate period after gene therapy. And we're still working out which gene therapy trial, which immunosuppressive regimen is appropriate for gene therapy. Next, is there an optimal stage of the disease to do gene therapy? Should we include patients who are relatively healthy and prevent the disease? Or should we include patients who have more advanced disease and hoping that it will stabilize their arrhythmia burden? or possibly also prevent further progression of the disease. Will we see a benefit, and how long will the benefit last? I think those are all still questions that are open. We have very encouraging data from preclinical models. However, the data on humans is still lacking. What we also don't know is whether redosing will ever be possible. So if we see an effect, but we see the effect for a couple of years, but then the effect gradually goes away, are we able to redose those patients with gene therapy in the future if they have already been exposed to AAV in the past? And the other most important question is, how will you, as our patients, will accept gene therapy? Have we done a good job of explaining what gene therapy is, what the safety and what the efficacy, and also what the potential side effects are? And just to summarize, so gene replacement therapy using AAV vectors is a very promising new therapeutic option, which has the potential to transform the management of many inherited heart diseases, including ARVC. And in order to do it right, we have to do gene therapy trials to better define the risks and the benefits of cardiac gene therapy. And with that, I want to conclude and thank you for your attention. And I've highlighted in red here the members of our ARVC team. Thank you so much for your attention.
Thank you very much, Dr. Barth. We appreciate that. We had one comment, question already in our chat field from the Netherlands, just asking if you're familiar with children who have ARVC or ACM. This person has had a son at 12 years old, survived a sudden cardiac arrest, and sees a Dutch pediatric cardiologist who is not familiar with these similar cases. We certainly can provide a physician referral in the Netherlands from the SADS Foundation. If you'd like to reach out to our sads@sads.org, we can do that. But Dr. Barth, would you like to comment on that?
Yes, so it's very important to see a specialized provider. And, within cardiology, we have pediatric cardiologists and we have adult cardiologists. So I'm an adult cardiologist. I see patients who are older than 18, 16, 18. There is sort of the border is not always that clear cut, but you have to be an adult to be seen by me. If you have a pediatric case, I would refer you to a very specialized ARVC center in the Netherlands. They will be able to guide you who the right physician for that condition is for this appropriate age group.
Thank you very much. Okay, we have another question coming in, and if you have other questions, please feel free to post them in the chat. The next one is from Heather. What if a person has two gene variants? In our case, a PKP2 and the DSG2 gene. Is this considered an exclusive factor?
Yes, unfortunately, at the time being, most companies would consider this as an exclusion criteria because the second variant just muddies the water. We wouldn't know is the effect that we're seeing or the lack of effect that we're seeing if you have two variants due to the second variant that wasn't treated. So we really want to include in this initial trials only patients who have one single PKP2 variant that is either disease-causing or likely disease-causing. If you have other variants, that may be an option in the future, but for now, you will be excluded from those trials.
Okay. And what is the reason that these clinical trials are only including people with ICDs?
It's a very good question. And it comes down to the safety aspect. Whatever we do, we want to make sure that you're doing well. And there is the concern when you infuse a large amount of virus that you elicit an immune response. And that immune response will lead to inflammation. When you have inflammation in the heart, you may theoretically, immediately after gene therapy is administered, see an increase in arrhythmias before you see an improvement in arrhythmia. So to mitigate that fact, we really want to make sure that patients take immunosuppressant medications to limit the amount of inflammation that the heart sees immediately after gene therapy. And to be protected, we want to make sure that patients also have a defibrillator. As you know, the defibrillator is really only the last resort. It's just a safety criteria that has been included, but we really don't want your defibrillator to go off in the first place. And that's the reason why we gave this gene therapy.
Okay, thank you very much. Any other questions or comments for Dr. Barth? Oh, let's see here. Here's one from Jason. How do you see treatment being provided? For example, would it only be provided to people with severe disease?
Yes, and I would, right now, we don't want to include patients who have only very mild disease, because likely we wouldn't be seeing a benefit. But at the same time, we don't want to include patients where the disease is too advanced, because we may not see a therapeutic benefit in those patients either. And it may not be safe if you're too far advanced. So right now, most trials will look at patients who are sort of in the intermediate stage of the disease. They shouldn't have heart failure, but some arrhythmias burden with ARVC is accepted. So most patients will have PVCs. It's not an exclusion criteria if you have had, for instance, a VT ablation in the past or ICD shocks. So that would still be acceptable. I think you're muted, Marcia.
I forgot. Thank you. Okay, a question from Jessica. What infections would cause our bodies to create AAV antibodies?
So AAV, it's a respiratory virus that often infects the body together with other viruses. So it's random. Any respiratory infection that has also AAV in the mix may create antibodies or may elicit an immune response. And it varies by geographical location, how many people will have been exposed to those viruses. It may be as low as 20%, but it may be significantly higher in some areas of the world. And this is also something that the companies are very much invested in to understand how many people with ARVC would be eligible for those gene therapy trials. Because if you have pre-existing antibodies, at this moment, this is a big exclusion criteria where we say, unfortunately, it would limit the efficacy of the gene therapy and would render it ineffective. So you would be excluded if you're found to have antibodies.
Okay, thank you. Heather comments, at the time we received the genetic test, the DSG2 was described as a VUS, a variant of unknown significance. Is that still the case, or is it now considered disease causing?
Yeah, so there are variants that are disease causing, and there are some that are variants of uncertain significance. There are some that are benign. And that classification is to some extent fluid over the years. So you will see over time that there are some variants of uncertain significance that get promoted to disease causing over time because we learn more information. But this opposite is also true, that we may have some variants of uncertain significance that are decoded, that we'll learn over time that they are actually not disease causing, that they are just background noise that occurs in the genome. And I would encourage you to contact your physician to discuss this specific variant that you're having with the genetic counselors to see whether there is more information that has been learned over time about this variant of uncertain significance. It may still be a variant of uncertain significance, but we may have learned more about your specific variant. And please discuss this with your genetic counselor that gave you the genetic test results.
Okay, thank you. Why are PVCs relevant for inclusion or exclusion criteria?
Yeah, that's also an excellent point. So we not only want to include patients who are genotype positive, meaning that they have a disease-causing PKP2 variant, but we also want to include patients who have manifest ARVC and to show symptoms, to show signs of the disease. Because if you're, again, too healthy, you're just a carrier of that gene, which can happen. You may be completely asymptomatic throughout your life. You would not benefit from gene therapy. And one of the signs that tells us, yes, there is some disease activity present, is the number of PVCs that you have on a 24-hour Holter monitor. So we use that as a surrogate of how active the disease is.
Okay, from Robin, how do companies decide which ARVC gene mutation to do gene therapy for?
Yeah, so it's when it comes to PKP2, there's an easy answer, because if you have all the PKP2 variants, the vast majority of the PKP2 variants that are disease-causing or likely disease-causing, they cause what's called a loss of function of the gene, which means you will express less protein of PKP2 if you carry this gene variant. When you have other ARVC genes, like desmoplakin, for instance, it's a little bit more difficult because they're not all maybe loss of function variants, and that's a very different discussion. So that's why it has been the choice to choose, to focus on PKP2 is because it's, well, the most common gene out there, so we find most patients. Their loss of function, so it's easier to interpret. And it also fits quite nicely within the packaging of this AAV gene. So not every gene may be suitable to packaging. So that made PKP2 a very easy candidate. When it comes to the other genes, ARVC genes, it may not be as easy. I can tell you that some of those genes are actively being investigated in preclinical models by various of these companies that I would encourage you to discuss that with them. But the data or the knowledge or the clinical trials are most advanced for PKP2.
Thank you. What impact does COVID have on AAV, the antibodies?
I'm sorry, can you say that again?
What impact does COVID have on AAV antibodies?
Yeah, so we don't think that there is a cross-reactivity between COVID or AAV antibodies. So COVID is a completely different virus. We know that COVID is a virus that can harm people. AAV is a virus that does not harm people. It can infect you. So that's what we use therapeutically, by introducing a gene into the cell without harming you. But it's not causing any disease. And I'm not aware of any cross-reactivity between COVID and AAV.
Thank you. Do you know if insurance companies cover the cost of gene therapy?
So if you participate in a trial, obviously the costs will be covered. And the expectation is later on, once these gene therapies are FDA approved, that they will also be covered by insurance. It will require a very rigorous pre-authorization process. But from other gene therapies, yes, they are covered.
Okay, thank you. Jesse asked, if I took a variety of antibiotics for an E. coli infection, am I disqualified?
No. If you use the antibiotics in the past, and just to remind people, so antibiotics are used to treat bacterial infections. That's very different from viral infection. But we don't want you to have an any kind of active infection, whether that's viral or bacterial or fungal, at the time when you receive gene therapy. And the reason for that is that we want to give you, or we need to give you, immunosuppressant medications in order to lower the immune response of your body towards the virus that you receive from the gene therapy. If you have an underlying active infection that is sort of just smoldering in the background, it may just flourish if you give you immunosuppressant drug and really become apparent. So that's why you really have to be healthy to be considered for gene therapy.
Thank you. Generally, when would you expect the therapies to be available outside of the clinical trial? 1, 2, 5 years or beyond?
I think within the next 2 years, if the gene therapy trials, go as expected, I would expect them to, to reach that level where we see them on the market.
Okay. So Carl asks, variants of unknown significance seem to be encountered in ethnic racial populations that historically have not been included in research studies. What efforts are being made to achieve greater diversity in these rare diseases?
That is an excellent point. And certainly, as you saw, the trial about inclusion criteria doesn't mention anything about ethnically any kind of ethnicity. We really welcome all patients with ARVC who meet criteria, irrespective of race and gender. The only criteria that we have right now is age, because the regulatory requirements for children are just the bar is set so much higher that we wouldn't be able to include children in the initial trials.
Okay, here's our last one, and then we'll move on to our panel overviews. Let's see. Just building on the question of the relevance of PVCs, so gene therapy could never be used as proactive preventative medicine, only reactive medicine once the genotype has started expressing via known phenotypes.
That is correct. What we know is that not every patient who has a disease-causing mutation will actually also show signs of the disease. We know that this is actually rare. Most patients who have that gene that disease causes will never show any signs. So to give them gene therapy would really not be safe, and it would not be effective, and it would be very expensive. So we really have to define the patient population who would benefit most from gene therapy. And for that, yes, you would have to show some signs of the disease. I think if you have a disease with very high penetrance, where you know, for instance, that if a baby has this specific gene, there is a 99% chance or 100% chance that this baby will also show signs of the disease later on in life, then you can think about preventive therapies. With ARVC, that is not the case. It's always a gene and the environment, and they work together. And there's clearly a role of the environment in ARVC. In some patients, the role of the environment is less. In some patients, it's a little bit more. But yes, we will need to show some signs of the disease in order to be eligible for ARVC gene therapy.
Okay, very good. Thank you so much. And thank you all for your questions with Dr. Barth. We appreciate it. And thank you for your time and expertise in sharing, Dr. Barth. So
Thank
we're
you
now, go ahead.
Thank you for inviting me.
You bet. So now we're going to proceed to our panel overviews with the 3 medical representatives from our companies here. And we are going to begin with Lexio and Megan Sutton. So Aaron's going to share the slide deck that you've provided. There we go. Okay, go ahead.
Hi,
Megan.
everybody. Can you hear me okay?
Yes, we can.
Hi, I'm Megan Sutton. I am a nurse practitioner and doctor of nursing practice, and I'm an executive medical director for clinical development and real-world evidence at Lexio. Before I joined the pharmaceutical industry, I took care of heart failure, heart transplant patients directly in Chicago. So I certainly have direct experience taking care of patients like some of you. So this is something that's quite personal to me. So it's been an honor to be a part of this. What I want to go over is just who we are as Lexio, talk about the gene therapy that we are doing with Lexio. Dr. Barth did a fantastic job really summarizing what gene therapy is, how it applies to people like you, and teach you a little bit more about our trial itself. Next slide. So Lexio is actually a company that's based out of New York City. And we have two major disease states or focuses that we're working on at our company. We are a genetic medicine company, so everything you see on this slide is gene therapy. And we kind of split into two categories. We have the cardiology piece, which you can see in the top. And in the cardiology focused programs that we have at our company, we actually have a trial that's ongoing right now for Friedreich's ataxia cardiomyopathy. So that's a heart disease that's caused by a gene. And the issue that these patients have is actually both musculoskeletal. These patients have a lot of symptoms like muscular dystrophy before they progress to the cardiac component. So we have a trial right now for gene therapy for those patients. And then we are working towards the PKP2 piece. You can see based on our clinicaltrials.gov listing that we have one of our sites open for our PKP2 gene therapy, and that's what we're talking about tonight. We also have two other things in our pipeline that are cardiac specific, and you can find some more information on our website. The other half of the company is really focused on the neurological piece. So you can see that we have a program for gene therapy and Alzheimer's, which is also can be gene created. So kind of two pieces to the puzzle when it comes to Lexio, but a lot of exciting things going on in the world of gene therapy at our company. Next. Thank you. So I understand that Dr. Barth went through this, and I have a feeling that a lot of my fellow colleagues from different companies are going to be kind of reiterating this as well. I think gene therapy is so new for everybody, patients and clinicians, and so there's still a lot of learning that has to happen about what it even is. And so what Dr. Barth said, as you can see here, the intention with our gene therapy, and this is what Dr. Barth mentioned, is the idea is replacing the dysfunctional gene that you may have as a patient. So what happens in PKP2 arrhythmogenic cardiomyopathy is there's a dysfunctional gene, a dysfunctional PKP2 gene. And when you have a dysfunctional gene, you lack the instructions that are required to build the proteins that are really needed by the cells. And so the intention here is to take that functional gene and kind of hide it within a viral shell. And that's what Dr. Barth was referring to as AAV viral vectors. And so what that means is you take a shell of a virus, you kind of scoop out the actual infectious part of the virus. And the reason why these are great is because viruses are very adapt at navigating your body. So all of us have probably had a cold at one point in time. So the viral shells are really good at getting in the human body. So we with gene replacement therapy can stick a functioning gene within this viral shell and remove the ability for you to get infected by this virus, and therefore it travels to the organ that needs to go. And that's what Dr. Barth mentioned when you're kind of choosing these viral shells. Do you want something to go to the brain? Do you want something to go to the heart? So the vectors that we use at our company are really designed to go to the heart, which is where this gene is intended to go. And what the hope is that once you get the gene within this viral shell to migrate to the organ that you need, it produces those instruction manuals to start to create the protein. So you'll see in a lot of the preclinical trials, these animal models that our companies have, you'll start to see evidence of those proteins forming. And so with the intention is once the proteins form, then you can kind of rejuvenate the healthy function of the heart itself. Next. Oh, perfect. This is a very detailed slide. So I want to give you kind of an airplane view into the point of this. What Dr. Barth mentioned is important. Where is the dysfunction with what's happening to you in PKP2? And so what happens in PKP2 is, as I mentioned, PKP2 is a gene that creates a protein. So you can see in the top left-hand side, patients that have an issue or a variant in PKP2 don't have the functional gene, and so therefore they don't build the functional protein. And what happens is this PKP2 protein is really a critical part in creating this bridge in between the cells of your heart. And so when you don't have a bridge, a really good bridge to be able to connect the tissues or the cells together, they can't communicate. And so the heart functions in two ways. It functions mechanically like a pump, it squeezes, and it also functions through electricity. And so when you have this dysfunction of the bridge, you can start to see those electrical issues as you can feel as a patient. And so the intention with gene therapy is to replace the functional gene that therefore can hopefully build a protein to build back that bridge. And so this is kind of a theoretical look into what the hope of gene therapy could be for PKP2. You can see on the top, this is what happens as a PKP2 ACM patient. You don't have the functional gene, therefore you don't have the functional protein, therefore you don't have that bridge that you need in between the cells. And that's what leads to the issues, that you're facing, this kind of change in the heart tissue itself, changing from a functioning heart tissue to more of a fibrofatty model, seeing that you might have arrhythmias, et cetera. And so what you can see in the bottom is when you add a functional gene, you therefore can then potentially create that functional protein to reform the bridge. Next. So this is an overview of our study. Our study is called the Heroic PKP2 study. As is the case with all of the companies, these are first in human studies. So you can start to see the terminology of phase 1, phase 2. What that means is that this is a first in human study. The previous studies that have been done are in animals only. So this is using our investigational gene therapy. It is administered through the IV. And everybody in the trial that is eligible that will get the therapy. You may have heard of other designs where there's something like a placebo or a sugar pill. In this particular trial, as of right now, there is no placebo or sugar pill. Everyone who is eligible for the trial will get this IV therapy. On the right-hand side of the slide, you can see some of the criteria that we're looking for in these patients. I know Dr. Barth mentioned in generalities, but you can see some of the criteria. This is not an exhaustive list, but it will give you a better sense of who we're studying for. I know there was a question earlier on about whether this is ever going to be considered as a prevention. I don't want to say yes or no to that question because I think that the hope is potentially a prevention. But as of right now, the way the FDA wants us to proceed is to study this in patients that have the disease. So you can see right here, we're looking for patients that are adults that meet the task force criteria, which means that they have the actual physical disease itself of ARVC. You can see that we want to make sure they have a documented variant of PKP2, that they have PVCs, that they have an ICD. They mentioned the LVEF piece of it, and you can see some exclusion criteria there as well. This is our last slide here. If you want any more information about this, the best place to find it is on clinicaltrials.gov. You can see that the number there, which you'll see in all of the trials, is important. So if you're looking for PKP2 trials, you can just type in PKP2 and it will give you the different trials. But most importantly, this is something that you really need to talk to your clinical team about, because there's a lot of information that all of these companies will not be able to provide to you tonight. And this is really a decision between you, your family, and your clinical team. So please, if you have any questions, talk to your clinicians about this. And I think that that's the best way to kind of proceed in asking and getting the information that you need as a patient.
Thank you very much, Megan. We appreciate it. And next, we're going to move to Rocket Pharmaceuticals and Dr. Jonathan Schwartz. And Aaron, you can advance. There we go. All right, Dr. Schwartz, you're up.
Hi, and thank you for the opportunity to share some information initially about Rocket Pharma and our commitment to patients and science, which is illustrated on the next slide. Essentially, all of Rocket's 300 some odd employees are deeply committed to developing transformative therapies that address serious disorders at the root cause. Backing up the talk are the events of the past 5 years, in which we have investigated 4 clinical stage programs, and together with our academic and other biopharmaceutical company partners have really been able to participate in ushering in a new era of genetic medicine. Over these years, we've treated more than 30 patients at 7 expert centers in Europe and the United States, largely all of them with highly fatal or highly morbid early life diseases. And all of these patients essentially are living now full and healthy lives. This year, we have 2 potential and in fact likely health authority approvals The first one of which will be in leukocyte adhesion deficiency 1, a very severe primary immunodeficiency, followed hopefully by another approval in Fanconi anemia, which is an equally devastating DNA repair and bone marrow failure disorder. 2 other programs have progressed to pivotal stage or registrational trials, meaning that if the trials meet the endpoints, we have FDA alignment that these would allow the therapies to become bona fide medicines. And that's in pyruvate kinase deficiency, another hematologic disorder, and in Danon disease. And the Danon disease program phase 1 results, as some of you may be aware, represent the first successful gene therapy for a highly aggressive cardiomyopathy. We have multiple patient testimonials regarding the gene therapy's effects on their lives. One of them is illustrated on the right portion of the slide in the cartoon bubble. For the sake of brevity, I won't read it. There are many others on our website attesting to the transformative potential of this sort of therapy in diseases like Danon disease, and with the hard work of us and our partners and fruitful collaborations, hopefully PKP2 ACM ARVC will be the next disease in which we're able to show such a benefit. When we started the Danon phase 1 trial, a lot of onlookers said, "Oh, you'll never be able to make gene therapy work in the heart. That's not going to happen." Fortunately, we didn't listen to them, but we did listen to a lot of other valuable advice from health authorities and from university and biopharma experts from around the world. And for that reason, we can attest to our track record of dedication to patient safety. In conjunction with colleagues at University of Florida and other institutions around the country and the world, we were able to help pioneer the use of the immunomodulatory regimen that's utilized in our phase 1 ARVC Pegasus trial. And that regimen was highlighted in a recent publication in the Journal of Clinical Investigation with co-authors from multiple gene therapy centers of expertise, in which we were able to demonstrate that this regimen markedly reduces the early activation of the complement system, which is one arm of the immune system that, when hyperactivated, can result in a very dangerous condition known as thrombotic microangiopathy, or TMA, and that by use of the immunomodulatory regimen that's in play in the current study, we're able to dampen that very significantly. Rocket's chief medical officer, which would be me, serves on 2 NIH-sponsored AAV safety working groups, and both myself and all of our clinicians and scientists are in frequent contact with multiple international experts regarding the safest and most effective ways of delivering gene therapy. Whether it's lighting up the Empire State Building and other structures like the Roman Colosseum in rare disease colors to increase awareness and funding, or working with any number of institutions and organizations to heighten disease screening and genetic testing and disease awareness. We have been relentless advocates for rare and high unmet need disorders. Moving to the next slide, I'll highlight some of the critical rationale that's underlying the RP-A601 phase 1 clinical trial. And really every aspect of this has been to develop an AAV vector and a clinical program that's designed to optimize the potential for safe and effective therapy. As is the case with the other programs, the gene of interest is encoded in an adeno-associated viral vector. And in this case, it's the full wild type gene. So in other words, it's the natural gene that people who don't have ARVC from PKP2 would have. And that gene is in a capsid that's an AAV serotype RH74, which we selected for this clinical trial after evaluating multiple AAV serotypes in preclinical studies in which the RH74 AAV demonstrated the most favorable safety and efficacy in relevant disease models so that we can give the therapy at doses that are likely to be effective and do it while having the optimal potential for safety. Importantly, the RH74 serotype has been evaluated in something like 100 patients with muscular dystrophy. Those are patients treated under the Sarepta program. And very importantly, that's a serotype that's not been associated with excessive complement activation and that thrombotic microangiopathy toxicity that I mentioned a couple minutes ago. And this is particularly important in a gene therapy trial in an adult population like ARVC, where you're giving relatively large doses, as high absolute doses have been hypothesized to predispose towards problematic complement activation. So this is really intended for both safety as well as efficacy. As is the case with the other trials, there's been very compelling proof of concept in a relevant animal model. In this case, we use the NYU model that Dr. Delmar and colleagues developed. And we were able to see that RP-A601 improved both the arrhythmia as well as the cardiac structure components of ARVC, as illustrated in the panels on the right portion of the slide. And very importantly, this AAV vector enabled long-term survival for multiple months in all of the treated mice with the PKP gene knocked out. The trial itself involves a single intravenous infusion, and very importantly, the starting dose for this clinical study of 8 × 10^13 genome copies per kilogram is one that we selected because it has a reasonable, in fact, very good probability of conferring clinical improvement. And we wanted to make sure, as we did with the Danon program, that we were entering the clinic with a dose that really had a very good chance of helping. The immunomodulatory regimen involves rituximab, sirolimus, and prednisone, and that is designed to minimize the potential for early complement and other immune-mediated side effects, and also to minimize the overall dose of corticosteroids and the duration to try to prevent the side effects that can sometimes happen from steroids like prednisone. In terms of what the clinical trial actually involves from a patient and study center perspective, the next and last slide illustrates a little bit of the procedures that are involved for someone who is enrolling and being treated on the clinical trial. It would start likely with events pertaining to eligibility and enrollment, with preliminary phone discussions and internet interactions, which, if suggestive that it's a good fit, would then result in an individual traveling to one of our study centers and engaging in initial eligibility screening and discussions with the investigator and study team. That would involve some of the genetic and antibody tests and a medical history, among other things. And that typically occurs about 2 months before one would actually receive the therapy. If eligibility is confirmed or considered probable, then there'll be detailed baseline testing at the study center, as illustrated in the lower left panel. That'll include heart tests, for which many of you are familiar with, as well as a heart catheterization with biopsy. And these are tests that are going to happen between 2 and 8 weeks before receiving therapy. If all proceeds well, then we would move to the middle portion of the slide, the treatment and initial monitoring phase of the study. One would receive intravenous doses of rituximab 2 and 1 weeks prior to RP-A601 as an outpatient, and then be admitted to the treatment center for a single intravenous infusion of RP-A601 with oral sirolimus and IV steroids that are then switched to oral prednisone. Typically, one is hospitalized as an inpatient anywhere from 4 to 14 days. And during that time and after, there's daily monitoring for safety. Predominantly, that's blood tests. But at times there might be EKGs and echoes. And then once discharged from the hospital, there are frequent outpatient visits initially, weekly monitoring for safety in the initial month, and every 2-week monitoring in the second months, again, predominantly blood tests. The goal is that all of the immunomodulatory therapy, the sirolimus and the prednisone, would be finished anywhere between 2 and 3 months after treatment. After that is follow-up, which involves efficacy and safety assessments every 3 months in the first year, every 6 months in the second year, which would typically involve Holter monitoring and echoes and electrocardiograms, with less frequent MRI and catheterization with biopsies. And then subsequent to that, additional long-term follow-up for an additional 3 years, with the emphasis there on less frequent and less invasive assessments. Hopefully this gives a good, reasonable preliminary overview. Of course, we'll have opportunities for further questions later in the sessions. And many of you are also familiar with Latika Hickey, who is our patient advocacy lead, who is always available to try to address your questions, help you work through any number of problems, even if you're not potentially a candidate for one study or another, or even if you don't have ACM, you have a different disorder. We are committed to trying to help any number of people in any number of ways. Thank you for your attention.
Thank you, Dr. Schwartz. All right, we're going to now move to Tanaya, and Dr. Whit Tingley will be presenting for Tanaya. Thank you.
Thank you, Erin. Thank you, Marcia, for inviting us. It's a great session. Next slide, please. So Tanaya Therapeutics is 100% focused on heart disease. Everything we work on is aimed at improving options for people with heart failure and cardiomyopathy, including inherited cardiomyopathies. And we do have 2 active ARVC studies now, an observational study called RIDGE and a gene therapy study called RIDGE-1. So we've built a team of people with passion and expertise in heart disease and cardiomyopathies. We have a genetic team that really looks through the most current human genetic research data to really understand what is causing and exacerbating disease in human people. And we conduct our own natural history studies with patients where patients tell us what they need next. We, of course, specialize in preclinical disease models in animals, and we have expert scientists committed to understanding cardiac physiology and pathophysiology in animals. We also have experts taking apart every aspect of gene therapy, the capsid, the cassette, the promoter, all the pieces you've heard about today. We take it all apart. We try every variation of every possibility. We screen for new variants, new approaches, and then we put it all back together in what we hope will be particularly safe and effective for patients before we bring gene therapies into clinic. We also do our own manufacturing. Gene therapy for manufacturing is a very complicated and very important process, and we feel the need to control it from beginning to end and hold it to the highest possible standards. We have 5 clinical studies now where patients are participating who have inherited cardiomyopathies, and we make it a priority to start talking early and collaborate long-term with experts at sites across the US and in Europe as well, both to design our studies and then to conduct them. But very importantly, our studies are designed with patient input and patient needs in mind. We have a dedicated advocacy team, including patients that have inherited cardiomyopathy, internal at Tanaya. We've built a patient board of advisors that comment on our trial protocols, inform consent forms, and really everything we do that affects patients. We do know that genetic testing is a barrier for patients really understanding whether they have ARVC and what form of ARVC, and we are working to develop no-cost genetic testing programs that will facilitate patient access to clinical trials. And we are committed to providing education, and I invite you to visit our website, arvcstudies.com. Next slide, please. Dr. Barth and other members of this panel have done an excellent job describing the disease and how mutations in the PKP2 gene cause a loss of PK2 protein or plakophilin-2 protein, and how that causes the buttons to unsnap. I love that analogy, Dr. Barth, of the buttons on the coat and what the desmosomes with PKP2 are doing. And this results in abnormal electrical connections between the cells and abnormal signaling between the cells and the cells pulling apart. And the result, of course, is ARVC. So the objective of what we're trying to do with gene therapy, in our case, it's named TN401, is to deliver a working copy of the gene to the heart cells to restore the normal protein levels of plakophilin-2 and restore normal heart function. So we're targeting the underlying cause of the disease in a way that no current treatments, devices, or procedures can do. And of course, to do that, we're leveraging extensive knowledge from successful gene therapies that have been developed in a variety of other diseases. Next slide, please. So we have selected to use the AAV9 capsid. Dr. Barth highlighted this capsid. We chose it because it comes with a robust validation, both from preclinical animal work and from clinical studies. AAV9 is the backbone for a gene therapy called Zolgensma for SMA, and it's been used in over 3,000 patients in many countries with long-term follow-up. In addition, there are already over 45 gene therapy trials using AAV9. So we understand AAV9 very well. We know what to look for in terms of the safety profile for AAV9, what to monitor, and how to react as the immune system recognizes AAV9. On the efficacy side, also AAV9 is validated in its ability to deliver gene therapy to the human heart in patients with an appropriate bias distribution, the amount of transduction or the amount of gene that gets into the heart, and durable gene expression over time in patients. We do know from head-to-head studies in preclinical models, including mice, that AAV9 outperforms other gene therapy capsids, and in particular drives a high expression of the gene, in this case PKP2. And it has the most extensive body of preclinical evidence for PKP2 disease models in mice. You don't have to take our word for any of this. This is all from folks in addition to Tanaya. AAV9 has been used in 3 independent preclinical studies of gene therapies for ARVC by different groups. Next slide, please. So the preclinical data for PKP2 gene replacement therapy is very robust, and you've seen examples across all of the sponsors. We are very happy with the way AAV9 works. Here's an example of how it improves heart rhythms in mice. So at the top is a typical EKG from a healthy mouse. In the middle in blue is a mouse lacking PKP2. And just like people with PKP2 mutations, these mice have arrhythmias, premature ventricular contractions, couplets, even non-sustained VTach. But when we treat with gene therapy, in this case, MTN401, which means the mouse version with the mouse gene of our gene therapy, we prevent all of these arrhythmias. We also restore the normal levels of PKP2 protein, and we prevent the scarring or fibrosis of both ventricles, the right and left ventricle, and slow the progression of the disease in the ventricles, and most importantly, restore survival to the normal lifespan of a mouse. Next slide. So our first in-human trial is actively recruiting now. It's called RIDGE-1. Our trial is very similar to all the other trials you've heard about today. We are enrolling adults with pathogenic or likely pathogenic mutations in the PKP2 gene and 500 or more premature ventricular contractions per day. An ICD is required, as are low titers of neutralizing antibodies. And Dr. Barth did a great job of explaining the rationale behind each of these criteria. The primary goal is to assess the safety of the gene therapy and to compare different doses. As you've heard with the other trials, we are starting with a dose that we are hoping will be fully active and effective. It's actually basically maximally effective in the mouse model. And we are not using a placebo comparator, so all patients in the trial are getting the active gene therapy. And of course, we are monitoring very closely for the effect on the symptoms and signs of the disease, including patient-reported outcomes. So the patients will be telling us how they feel after the gene therapy. Our first 2 clinical trial sites you'll see on clinicaltrials.gov are Mayo Clinic and Johns Hopkins, and we will be announcing other sites very soon. Again, we are choosing sites based on their expertise in ARVC. Please contact our patient advocacy team to learn more. You can email at patientadvocacy@tanayathera.com. Our website is arvcstudies.com, and we are on clinicaltrials.gov with the NCT number that is shown here. Thank you all for participating in this. I'm glad you're interested in participating in helping SADS. I hope you also contributed with the patient-focused drug development meeting. That helps us. I know it helps the FDA as well. And please reach out to us to give input. Again, we very much value input from patients as we design and conduct the things that we do. So thank you.
Thank you very much, Dr. Tingley.